welcome to the Rising Star session. My name is Dragan, and I'm happy to welcome also Tamas from 45 Ventures. So the procedure is as following. We have 1 minute of intro and transition, 3 minutes sharp of a pitch, and 1 minute for Q&As. Let's get started with the first project, Modiblast. And we are going to learn something actually about Trojan horses, which is very interesting. So it's about cell programming regiment, turning cancer cells into antigen-presenting cells.
Okay, thank you very much. My name is Helga Schmetzer. I'm the founder and CEO and CSO of Modiblast. We developed in vivo personalized cancer therapies. This means we use a novel concept to reprogram cancer and immune cells in vivo. With our patented strategy, we active both the innate and the adaptive part of the immune system and generate leukemia-specific effector and memory cells. Our initial target is the blood cancer, especially acute myeloid leukemia, which is the largest market in leukemia. In long term, we aim to create administration options that enable patients to dose themselves at home, potentially in several different treatment options. Our strategy combines two factors our body uses to control the fate of immune cells in one drug. A differentiation factor that converts stem cells into immune cells, alerts the immune system and supports hematological recovery. B, a danger signal which promotes the maturation of dendritic cells. This combination can reprogram cancer cells directly into dendritic cells of leukemic origin, or DCLOIs as we call them. In other words, we turn the foes into friends. These DCLOIs carry the patient's individual cancer antigens and thus can reinduce personalized immune responses. In the interest of time, here is a short overview. Numerous preclinical in vitro and in vivo results show that a multitude of leukemia-specific immune cells are generated and immunological memories established. In animal models, our lead compound was safe and reduced blast counts. These effects were observed in blood and in bone marrow. We also expanded our clinical network and gained early human data from off-label uses in advanced AML patients. We are in the process of publishing this data. Overall, our data is shaping us to be a comprehensive IND-enabling package, also reflective by a positive BfArM review. We are currently in discussion with investors for the upcoming financing rounds in two steps. A seed round with around 3 million euros will enable us to take our lead program to CTA submission. Further in vivo data and data on tolerability should pave the way to the clinic. We also aim to expand our patent position and generate regulatory value, for instance, by securing orphan drug status. A Series A round of roughly 11.5 million euros should get us through the planned phase 1/2 trial. We see clinical proof of concept as a key value inflection point. In summary, why is Modiblast a promising investment? Firstly, we've shown significant progress towards the clinic with a clearly differentiated strategy in AML. Second, as an in vivo personalized approach, we avoid many problems cell and gene therapies face. We have no complicated logistics, no cold chain needs, no long waiting times for the patient. Third, our approach combines the best of two worlds, a personalized and yet off-the-shelf cancer treatment. I thank you very much for your attention, and I'm happy to address any questions from your side.
It's a pleasure to be here back, actually, because we've presented some 5 months ago, so I can give you a little bit of an update. So my name is Slim. I'm a medicinal chemist by training. I'm also the co-founding CEO of PROSION Therapeutics. We are a German spin-off and we hold very proudly the Seal of Excellence of the European Commission, as we have a platform technology to basically dive into the undruggable. More specifically, we are targeting protein-protein interactions as they orchestrate any type of cellular malfunction leading to disease. Why is that important? Because they are known to be holding the next treatments for human health, and current methodologies and weapons we have either cannot reach protein-protein interactions, or just like small molecules are not complex enough to go for these type of interactions. If you dive even deeper into protein-protein interactions, you'll find out that there's one class that is extremely abundant around the recognition of proline-rich motifs, and some of the most notorious undruggables out there use these type of interactions in their roles in cancer, Alzheimer, and heart disease. Just to name drop a few, MYC-p53, they all use the recognition of PRMs in their activity, but they're still undruggable, and that's exactly where we come into the picture. We have found a way to control this type of interaction through our PROM technology. What are PROMs? Proprietary technology by PROSION that allows us to develop synthetically new molecules that are on an uncharted chemical space able to copy the helix and the conformation of these type of interactions. The way we use them is we combine usually two of our PROMs into something that is small enough to fit into the cell, but also has the specificity of a biologic in order for it to undergo interactions with these type of protein-protein interactions. So that's really how we'd like to position our technology and how drug hunters like to say it and see it. It's basically bridging the gap between small molecules and biologics, as it's bringing the advantages DMPK and also cell permeability of small molecules with a specificity, but also the target broadness of biologics. Now, it's great to have a technology. It's even better to make it be useful and to use it in cases. We have an entire case study that shows you basically from simple idea to in vivo proof of concept that we can tick every box around the way, from MOA, but also on target specificity. And we are currently focusing a lot on our lead program, YAP-TAZ, known in oncology for its role in the Hippo pathway. It's a highly validated target, so for us, low-hanging fruit. It's obviously undergoing proline-rich motif-mediated interactions, and there is already a lot in the clinic showing that the Hippo pathway itself is highly well tolerated when it's drugged, but current attempts are going more towards TEAD, which is showing great tolerability, but efficacy currently only in mesothelioma. There's data showing that YAP can go beyond that and be active in much longer lists of solid cancers, as it's basically translocated into the cell nucleus, and that's exactly what is driven through its PRM-mediated protein-in-protein interactions. That's great, that's where we address, and that's exactly where we have developed the first binders to it, and we have new data to present, obviously under confidentiality. We're currently raising our seed round, as we have a lot of non-dilutive funding also in the back, and I'll add here with a little bit of a picture of the pioneers behind this innovation, combining the experience of biotech, but also smart big pharma thinking and maneuvering. Thank you very much. Looking forward to maybe one question or not.
Exactly.
I work for Akribion Therapeutics, and we have set out to create a completely new class of treating cancer using our programmable cytotoxic nucleases. Now, how does that work? What is this? First of all, our technology, which we like to call our molecular shredder, consists of three core components. Firstly, the enzyme itself, which is a nuclease that shreds all DNA and RNA within a cell until the cell dies. But that only happens if an activation occurs between our so-called guide RNA, what we call Seekyo, and the cellular RNA that we can program our nuclease against. The third component to complete the system is a delivery system, which we are partnering with well-known companies and are using LNPs. The whole system is patent protected, and we have a granted patent in Europe. Now, let's have a look at a little bit of data. What you see here is a co-culture between healthy cells in red, HEK cells, control cells, and cervix carcinoma cells in green. The green cells carry an oncogene E6, which is one of the oncogenes for HPV. Now, if we put GDSE in there without a guide RNA, nothing happens and the cells survive happily. If, however, we put in a guide RNA that matches the RNA of the oncogene E6, you see that almost all, or in this experiment, 98% of the cells died, and that has happened over and over again, no matter which target we choose. However, we've not only worked on that one experiment, we have identified so far over 20 different indications that we will be able to target with our nucleases. That, of course, also leads to very substantial markets with the whole platform, from our perspective, having a potential for a multi-billion-dollar revenue in Akribion. In total, there's 4 key USPs we offer. First of all, we can address a completely new target class, RNA, that is currently not addressable as a biomarker. Secondly, it's extremely easy to reprogram. Just change the guide RNA and you can target a new cancer. We have it patent protected and we see very low off-target effects so far. Now, the team consists of myself and my co-CEO, Michael Krohn, the former head of R&D of Brain, along with a very strong management team, and we've been working together for a few years already. Currently, we are preparing our seed financing round. Actually, we are pretty close to closing that with the main goal of achieving an in vivo proof of concept for our lead indication, which is HPV positive head and neck cancer. For our second indication, which is solid tumor liver metastases that originate either from the colon or the pancreas. And thirdly, to start aligning with regulatory authorities early in order to make sure that we are well prepared for what is ahead. For that, we are hoping to close within the next couple of weeks, actually, in October. And the syndicate is currently at the round is supposed to be at 8 million, and we are currently oversubscribed. Now, let me summarize. What should you take away from this? This is a completely new technology. We can address new targets that no other company, to our knowledge, can do. We are extremely easy to reprogram to attack a different cancer or even a different cell. We have patent protection on our technology. And last but not least, the low off-target effects we consistently see in our experiments are a good indicator that this will actually be a technology that can move the needle for patient benefit. Thank you very much.
So how do you ensure that you get persistent RNA degradation of your target RNA?
Actually, it's not just the RNA that is being degraded. The RNA is just the activator. Once the activator once a match has been made, the enzyme undergoes the conformational chains and chews up all RNA and DNA. And if your question is specificity, we've created a lot of data, which we can unfortunately only share under NDA, that indicates that a singular mismatch in the seed region of the guide RNA already prevents an activation of the enzyme. And if there is one mismatch towards the very end of the guide RNA, that might be tolerable. However, when we design our experiments, we can ahead of time always make sure that there's at least a 5% difference between our target RNA and any potentially occurring RNA within the cell.
All right, thanks a lot. That's our company in a nutshell, and let me guide through it. So we are ABYOLO. ABYOLO is a company which is driven by purpose. We want to make biological medicine accessible and affordable to everyone, everywhere. What does ABYOLO stand for? ABYOLO is affordable biologics because you only live once. Now, we are talking in biosimilars about a market which is growing over the next years up to $126 billion market. Until 2033, the blockbuster sales volume, which is running out of patent, is totaling to $265 billion. In our first year, we are starting with 2 biosimilars, and every other following year, we are adding 1 biosimilar. Now, our first biosimilar, we all have the technical details. We have everything piled up. We are ready to go. And we have a kind of a proof of concept for that as well. Our pipeline, we run through a complete selection process with various attributes which we took into account, whether it's launch timing, partnering, or competitiveness, etc., etc. And it consists now 7 biosimilar candidates, most of them oncology, immunology, biosimilars. Nevertheless, we are starting with a target market of 10 billion in hematology. Now, our team consists of a total of 4 persons, 2 experts in the biosimilar field who are veterans. So we have Professor Niazi. He's a US-based professor who founded the first biosimilar company in the US. And we have Professor Omastotir, who runs several biosimilar projects from the beginning to the end. The team really has an extensive experience, as I said, and Professor Niazi is also driving the regulation in the US and in Europe as well. He was also part of the advisory with the Obama administration. Now, in addition to that, we have 2 business people who ensure executing on our mission. What is our USP? Our USP is that we can leverage from all the new technology which is available. It's automation, it's robotics, AI, and ML. We can increase our throughput by 3x compared to our competition, and we need 70% less personnel. So if you have an existing company which needs to kick out 70% of the personnel, you can guess what kind of exercise that would be. We also don't have a legacy, and legacy in companies and corporates look typically like this, very split, right? So we are starting from scratch, and that allows us to leverage everything. We don't need to write off existing assets either. So you have a strange discussion with the CFO, possibly, if you go to them and ask for that. So we are looking for 44 million euro, as I said. It also includes already from beginning to the end for the first molecule, from which we expect the first revenues coming in the first quarter of year 4. As of year 7, we are profitable. We are dividend play. We pay out the dividend already. And year 10, which is our perpetual year, we are going to pay out every year 3.2x dividend of the total paid-in capital. And with that, thanks a lot, and ready for the Q&A.
AIPTO TechBio is a TechBio startup in Basel. Our mission is to cure brain cancer. As we all know, brain cancer, including primary tumors and brain metastasis, is a devastating disease where we don't have many options. Now, the biggest challenge is that the current preclinical models do not translate to the clinic. The human brain and the brain cancer is so complex. The current models fail to capture it, not mention the heterogeneity of patients and tumors. Now, AIPTO, our TechBio startup, address this problem with a brand new approach. Our patent-protected next-generation patient avatar technology. Instead of dissolving the tumor tissue into individual tumor cells, we actually keep the tumor tissue intact and grow the intact tissue in the lab as if it's still in the body. This breakthrough technology maximally maintains the tumor microenvironment and can predict how patients respond to the treatment that no one ever has been able to do before. In addition, we can perform single-cell RNA sequencing on the lab-growing tumor avatar. So we capture single-cell level drug response, creating massive single-cell data that's clinically relevant, enable us to train generative AI models that will scale up the prediction and reveal the underlying mechanism of resistance and sensitivity and so on and so forth. This technology, this breakthrough approach, have been fully validated. We have successfully cultured patient avatar for more than 300 patients. The avatars, the tumor avatars grow in the lab, diagnostically is indistinguishable from the original tumor. We have shown our foundation AI model can indeed predict drug response on the single cell level. In a clinical study of 22 GBM patients, we show that our technology can predict patient survival, outperforming the current biomarker by a large margin. Now, what does this all mean? Combining the patient avatar in the lab and generative AI in the cloud, we have a predictive technology that's unprecedented, and it will impact every step of drug discovery and precision medicine. And we aim to leverage this to innovate and develop and co-develop novel, effective, personalized brain cancer therapeutics for the patient. We have already validated novel targets and molecules, and we have ongoing collaborations with pharmas across all the modalities. We have a team of experts. The patient avatar technology is coming from Professor Heiko New's lab in German Cancer Research Center. Myself is coming from AI background, leading AI for drug discovery group in Novartis for 10 years. And we are raising seed fund and looking for all collaborations as well. Please join us. Thank you.
Thank you for the intro. So good afternoon, everyone. We're looking to develop innovative drugs for rare CNS disorders. Actually, the PROTIDE technology was developed to actually deliver unstable nucleotides in intracellular targets. And basically, the chemical branches that are added make the active drug substance inert all the way until it reaches target site. Now, what this does is it changes the entire PK/PD, and therefore you can get an NCE patent. Now, this was tested in sort of HIV, etc., and in cancer, where you can see a 200% increase in the amount of active substance delivered to the CNS. So we are taking this across to actually develop to develop drugs to actually target CNS disorders, starting with rare and then probably looking at even non-rare indications. So why rare? We're looking at Niemann-Pick type C and Batten. I think the team, we have the leading researchers for lysosomal storage disorders in the team, as well as my lead founder, Stefan, who knows every single KOL one-on-one in the world in these disorders. Now, what we see here that both these diseases are extremely debilitating with mortality happening within the first 3 decades of life. Now, Miglustat, a sugar analog, has shown slight improvement in efficacy with extending survival by about 10 years. But because of side effects, the dose has been extremely limited. So what we did, so we looked at our technology and used a PROTIDE Miglustat to actually see that there was a 3 to 5-fold increase in the reduction of lysosomal storage volume and material. We activate the TFEB nuclear translocation, and at the same time, we are avoiding all the side effects which had a dose-limiting impact on Miglustat. So what that does, essentially, as you can see, is in the NPC mouse model, the dose used was 150 milligrams per kg, while in humans, the approved dose is only 10 milligrams per kg. Now, using our technology, we can avoid all these side effects and actually hope to increase the efficacy just so that we can see a survival that is going to be similar to what we see in the NPC mouse model. Now, as you can see here, we've also done, this, someone would say, why rare diseases? But I think it's a very healthy opportunity. It has an opportunity to have about $500 to $600 million sales worldwide, and the opportunity to kind of extend that as we move it to other indications. It also, we kind of proof tested it, and it has a risk-adjusted NPV, even at a 25% discount rate, that is positive. So we're looking for about, €1.5 million to bring these two, get INDs for the 2 indications. And again, why we are saying us, we have the knowledge, the network, the experience. We have a clear scientific rationale, a 14 months to IND, and we have an exponential business potential by extending it to GM1, GM2, etc. Also rare diseases in the CNS. That's it. Any questions? Happy to take any questions.
Thank you very much, and good afternoon, everyone. I'd like to introduce you to a fairly new and emerging area of biology called tRNAs. I'm CEO based in Zurich, where the company is based in Belgium, and we're developing a new class of therapies, small molecules, which are modulating the expression of tRNA. So cancer immune evasion is the big problem here, right? And we know this, that cancer is able to get around the immune system and avoid immune recognition and destruction. So how can we evade? How can we change cancer from the inside rather than addressing it from the immune system? So we're developing now a new therapy or a new class of therapies called tRNA modulators. They are acting on the protein expression machinery and modifying how much protein is translated. And this directly affects how cancer grows, how it metastasizes, and secondary, that you can see on the right side here, is the immune activation, and this is the CD4 cells in a co-culture model acting on the cancer itself. So what is tRNA? tRNA transfer RNA is again quite a new area or emerging area of biology. It's taking the mRNA and translating this into the protein formation. And what happens here with tRNAs are, if you interfere with this process, you essentially get a mistranslation of the proteins, and you also get new tumor antigen formation. So we can imagine it, let's say on the left side here, this is cancer. This is the car driving at full speed on the highway. Okay, you get enhanced translation and you get immune evasion. So the high-speed cancer needs more and more proteins to grow, to metastasize, to spread. And tRNA modulators are essentially putting a cruise control in this, slowing it down. It's maintaining the normal cell function of the body, but it's creating suboptimal translation. So those proteins are not able to get to the cancer to feed the cancer's growth. And the new tumor antigen presentation stimulates this new immune activation of the therapy. So these small molecules are having almost a genetic type of phenotype. We've developed a platform technology that enables us to identify novel tRNA targets, to validate those using in-house technologies, and we have applied AI computational platform which we have in-house to position, to identify cancers where the certain tRNAs that we are targeting are highly expressed or overexpressed, and how we can identify novel indications. We are looking, we're just finalizing now a seed extension round. We are looking for 5 to 7 million to finalize the IND studies for our first target. So this is not, this is a proof of concept in melanoma, but the final clinical indication will be one of several that we've identified. We currently have a 3 million committed from existing investors, VCs, and we would be happy to discuss with you if you're interested to participate and learn more about tRNA biology. Thank you very much.
Thank you for pronouncing my name properly, because it's rarely I hear that.
I was practicing.
And NatIgGs, it means natural antibodies, natural available therapeutic immunoglobulin gamma. We specifically address or focus on the proteins misfold and cause the disease like Alzheimer's. We are raising seed fund. The amount is exactly not clear based on the collaboration. So what we try to do is, like, we have a lead and we would like to optimize, and we would like to go for the right model in the animal model, which is not the mouse, but it's a dog model, so it's kind of pricey. We have a team of 4 full-time employees covering all different disciplines of biology. And it's based in Ulm, Germany. And the proteins misfold in different organs, not only in the brain, not only in Alzheimer's. So we built an antibody rediscovery platform, which could be then translatable to virtually any misfolded proteins that could be in the brain, heart, or whatever. Let's understand the protein misfolding with the infamous, Alzheimer's, the peptide from hell, amyloid beta. The monomers, I'm talking about the clinical trial phase 3 data. So anything that targets the protein as monomer, the amyloid beta monomers, never work. Anything that touches the fibrillar form in the plaques always creates some adverse effect, like amyloid-related imaging artifact E, edema, or hemorrhage H. And what we need to target is oligomers. Everybody who worked with Alzheimer's disease, they all talk about oligomers. Number of antibodies that ever entered the clinical phase, 0. And that's what we try to address. The problem is, we all know that the oligomers are toxic, but we don't know the shape and the conformation, the structure of these oligomers. And that's why we need to rely on our best partner, natural antibodies. We are born with good protective antibodies. We don't have Alzheimer's when we are 20, 30s, because these antibodies keep us healthy. When this antibody goes down in quality and quantity, the protein misfolding kicks in. And that's why the Alzheimer happens. So if you keep this antibody supplied, so identify the right protective antibodies, technically these are highly specific, but low affinity. That's why we use this buzzword AI, and the computational approach to optimize the antibody's properties, and then we simply resupply them. And this is the evolved precision, because we are talking about protein that misfold, which is in our body, and they misfold, and we have to rely on the evolved precision with the natural antibodies and using the cutting edge technology AI, then we can have this therapeutic molecule. As I mentioned, we have a great team of biochemists, biophysicists, computational biology, cell biology, and everyone having PhD, different nationalities. I think this is the dream team, a bragging slide, let's say. And we would like to go from the lead optimization to the KNN model and then goes further. And these are the scientific network collaborators and support team. I have 10 seconds. Thank you so much.
Hello, good afternoon, everyone. My name is Annegret van der Raa. I'm CEO of Cantoni Therapeutics. And today I'm presenting you what we are working on, NNMT, a novel and a promising target for cardiometabolic disease. So as you may all know, cardiometabolic disease is a major healthcare issue today. It is actually the #1 cause of death, causing 20 million people dying every year, due to a range of comorbidities, including diabetes, atherosclerosis, and obesity. Current treatment options for the disease are definitely available, but a significant proportion of the patients is suffering from either not tolerating the drugs, experiencing side effects towards the drugs, of not having any clinical response at all. We also know that due to our current lifestyle, the population of patients will increase with cardiometabolic disease, and so will the market. Therefore, we believe that there is a high unmet need still for new, effective treatments in this disease. How do we want to tackle that? This is through a novel and promising target called NNMT. NNMT stands for nicotinamide N-methyltransferase, and it's actually a metabolic enzyme that was discovered in the late 1950s by an Italian chemist named Giulio Cantoni. The enzyme is predominantly expressed in the liver and in fat tissue, and the enzyme is actually a central regulator of energy expenditure, regulating also the expression of NAD+, and thereby impacting both glucose as well as lipid metabolism. And you can imagine that such an enzyme in prehistoric times played a pivotal role. After the hunting, the NNMT levels in the human beings went up, and that actually caused energy to be stored and could be used in times of scarcity. However, today, this is not applicable anymore because we have an abundance of calories continuously available, and we see that patients with cardiometabolic disease, in fact, have those increased levels of NNMT. Therefore, at Cantoni Therapeutics, we are developing best-in-class small molecule inhibitors towards this metabolic enzyme, and we have identified highly potent and selective compounds in vitro, as you can see it from the biochemical NNMT assay at the top. And this in vitro potency is actually translating into an in vivo effect as well in relevant animal models. So on the one hand, we have dosed for 4 weeks diet-induced obesity mice with our compound, and we see combined effects of reduction in fat mass compared to the non-treated animals. We also see a reduction in blood glucose towards normalizing levels, and we also see a decrease of insulin resistance in the model. Moreover, what we also have done is treating in a golden standard humanized dyslipidemia mouse model with the compounds for 16 weeks. And there we see that the lipid reductions that we measure, that they are actually preventing the atherosclerotic plaque to be developed. What we have as investment opportunity at Cantoni Therapeutics today is therefore best-in-class NNMT inhibitors, which show significant potential as a novel treatment for cardiometabolic disease. With the company, we are currently raising 15 million euro, and the proceeds will be used to execute the preclinical development and the phase 1 clinical development plan, including patient cohorts. So we are offering an exciting front row seat investment opportunity where we are aiming to get clinical data available by 2027, and where we know in the meantime from further research that NNMT also has potential in other indications beyond cardiometabolic disease. So happy to take any questions, and do not hesitate to contact me afterwards if you want to know more. Thank you.
Hello, everybody. I'm Zaki Selam. I'm very happy to present you today. Just want to make sure I see the screen. Roca Therapeutics, which is developing first-in-class medicine against severe ocular disorders. So to start with an introduction about the company. So Roca Therapeutics is a spin-off from University of Nice. The company is currently VC financed thanks to the support of 3B. And the company has been created in 2021, trying to exploit a science around alternative angiogenic pathways. Everything that is related to neovascularization that is subject to resistance is the type of science that we've been addressing into the company. And actually, we have been able to develop in a couple of years and select a lead clinical candidate named RCT002, which is at 12 to 18 months from the clinic. And we have this compound protected by a patent composition of matter. And we are looking right now for the Series A financing, co-led ideally by our current investor 3B, to be able to further develop this asset up to the end of the phase 1/2A. And the team is based on a combination of experienced industry experts, including myself. So I'm myself the president of the company. I have more than 25 experience in the biotech arena, and also a brilliant team of complementary academics with expertise in the biology of neovascularization and in the medicinal chemistry. So in terms of background science, what I can tell you is that if you might have heard about the main treatment which are used right now for all the retinopathy and ocular disorders are the anti-VEGF. And basically they are given to patients using a needle in the eye, and that's the main standard of care. And that's an established one. The issue is that when you get to treat patients with anti-VEGF treatments, you have, first of all, a significant percentage of the population that does not respond intrinsically. And then you have a majority of patients that after sometimes become resistant. Why? Because there is a mechanism of resistance that is being developed. And one of these pathways that is mediating that resistance is the one that is named ELR plus CXCL, which are a number of cytokines. The most known is interleukin 8, which binds to CXCL. And the issue with that pathway is that it's not only an alternative one, it continues to sustain neovascularization that leads to resistance to anti-VEGF treatment, but also to sustain a chronic non-resolved inflammation. And within Roca, we decided to address that pathway as a way to overcome resistance to anti-VEGF treatment. So if you look at the issues with retinopathies and ocular disorders, the main issue is that you have a combination of multiple pathobiologies. First of all, you have inflammation, which is addressed right now with corticosteroid. But the issue is that corticosteroid can't be used, forever. They are broadly non-specific, immunosuppressive, and for a significant number of patients, it leads to cataract. And then you have the angiogenesis, which is well addressed with anti-VEGF, but leads to some resistance that can't be addressed. And then you have fibrosis and oxidative stress. We at Roca Therapeutics decided to address these four pathobiologies in a concomitant basis. And actually, we developed a selective CXCR2 antagonist, which is addressing this pathway, and a DRAG1 also antagonist. And we've been able to get a number of data which are extremely encouraging, showing that we have a compound right now that can be used on a non-competitive basis, but really complementary basis to anti-VEGF, being administered through the eye as a topical administration. We've demonstrated safety, tolerability, efficacy, and bioavailability through the eye in multiple spaces. And with that, we decided to be focused, first of all, on an indication in which we could have the orphan drug designation, so that we could get rapidly into patients without having to go to healthy volunteers, and to be eligible for early approval. And just as a summary, this is a program that has a unique pharmacoeconomic profile because it is a small drug that can be administered through the eye, complementary to the anti-VEGF, accessible for the patient's resistance in countries that do not have access to anti-VEGF, and also potentially disease modifying due to the mode of action by itself. And with that, I would like to thank you. Sorry.
First of all, thank you very much for this opportunity to present FluoSphera today. So I will start with the bad news because, as you know, most of drugs entering the clinical trials fail because of unforeseen toxicities and lack of efficacy. And our mission at FluoSphera is to change the paradigm in drug discovery by providing human and systemic in vitro solution to much better predict what is the effect of drugs, and so to bring safe and efficacious drug to the patients. So to do that, we invented a technology of tissue multiplexing, allowing us to mimic the systemic organization of the human body. And in addition to that, we also develop additional technologies. And thanks to that, we can propose to our customers an integrated set of solutions that can de-risk much better the drug discovery pipelines. So first, we have a solution to better predict the selectivity of drugs. We can multiplex up to 7 different tissues to make the comparison with an unbiased manner. Then we have a second product where we assess the safety and the efficacy of tissue in systemic condition, meaning it's the presence of the liver that can metabolize the drug. Then we have a third solution where we couple that with omics analysis to identify systemic biomarker, and the last one is to identify the most relevant animal species for preclinical studies. So since we favor the discovery of NCs and NBs, we impact a market worth $500 billion. And of course, FluoSphera is targeting a market a bit lower, valuated approximately to $3.2 billion. And so today, two things are totally impossible to combine: the high throughput required for drug screening, and then the systemic relevance to de-risk before the clinical trials. And typically, the systemic relevance is achieved through the animal models during the preclinical studies. And at FluoSphera, we can combine the high throughput with the human and systemic relevance. So we address really specifically a niche in the drug discovery market. So to capture the full value of this offering, we have a business model based on drug discovery contract with research milestone, and then we provide data to customer through commercial license agreement that are bound to the payment of success milestone and royalties if the drug is reaching the market. Since the pre-seed round that we raised in 2023, we continued the development of our technology. We filed additional patent, and now we are signing a contract with big companies. Also, we have some recurrent customers, so meaning that there is a true market value of what we propose to the market. And now we are specifically targeting the market of antibody drug conjugate, because many of these drugs are failing in the clinical trials because of systemic toxicities. And this is exactly this need that we address. And recently, we integrate the top 100 startup ranking. So we have an amazing team to develop this technology platform with my co-founder, Clelia, who was performing drug repurposing at Novartis. Also, we have as a chairman, Igor Fish, who also is a serial biotech entrepreneur. And Jean-Pierre Penassino, who is the CFO of MedAlliance that was sold to Cordis last year for $1 billion. And so now we are raising a seed round of $3.5 million to develop our technology platform or business
to become a market leader in translational business, and most importantly, to provide a patient with safe and efficacious drugs. Thank you very much.
Welcome everyone. I'm Jan Nilis and I'm currently the project lead here at Ariya Bio, which is a proteoglycan spinning out of ETH Zürich from the laboratories of Professor Jacob Korn, who's a worldwide renowned expert in gene editing. And what we aim to do at Ariya Bio is we want to turn blood stem cells into living protein drug factories to cure rare diseases. And as you are probably well aware, today there's still hundreds of millions of patients affected by rare diseases that lack an FDA-approved treatment. And we want to change this. So what we do is we start by extracting the patient's own blood stem cells, also called hematopoietic stem cells, and gene correct them using CRISPR-Cas, then reinsert them into the patient where they home to the bone marrow and stay there for the rest of the life of the patient. And then these stem cells will form different kinds of downstream cells, including, of course, T cells, B cells, but also red blood cells and red blood cell progenitor cells. And those are the ones that we're really interested in. So what we are doing is we're reprogramming these blood stem cells so that we can express cargo, therapeutic proteins and peptides from these red blood cells and the red blood cell progenitors. And then we leverage the body's own delivery system, the blood, namely the red blood cells, to deliver these protein drugs and peptides throughout the body via the vasculature. And we believe this is really advantageous as blood cells in the vasculature can really reach every single organ in the body. And also they're programmed to produce large amounts of proteins. So if we can only hijack a small fraction of that, we can produce large quantities of protein drugs directly where they're needed in the body. And also whatever cargo you put into them will be tolerated by the immune system. So now this is not so easy, as also red blood cells are infamous for basically silencing everything that's not a hemoglobin gene. So how do we get around this challenge? So what we've developed, we refer to as hematogen, and it's actually quite simple in essence. It's a two-component system. Firstly, a new set of synthetic promoters that we have developed that stay active in red blood cells. And secondly, a novel safe harbor site that is also a globin site, which is also known to stay active, but it's not essential. So we can insert any therapeutic protein into there and express it lifelong at high quantities. And we can easily reprogram this technology for any other indication that we would like to target by simply swapping out the therapeutic gene. And also, we're always reusing the same guide RNA and the same nuclease, so we always have the same off-target profile and can cross-reference our off-target profiling in any further IND application. And yeah, CRISPR components are going to be off patent soon. So far we've raised 1.2 million non-dilutives, which has enabled us to validate our promoters. And we've also published our work on these promoters in a peer-reviewed journal. And we've also started validating our full gene size insertion workflows and are getting really high insertion efficiencies already. And we've got a patent application pending. So now we're raising a seed round of 3 million Swiss francs, which aims at taking our first asset, which is in pyruvate kinase deficiency, all the way to pre-IND meetings. And this means in vitro and in vivo proof of concept, as well as, of course, first safety and safety package and a GMP-ready clinical protocol. And also we'd like to then, with that money, also perform 2 in vitro proof of concepts, 1 in hemophilia and 1 in POMP. And we've also got sickle cell and thalassemia programs, which we're not actively pursuing, but open to exiting them if there's an interested partner. Yeah, last slide. So we're spinning out of the laboratory of Professor Jacob Korn, who's among the CRISPR-Cas pioneers, who has also founded or was heavily involved in the founding of the IGI in Berkeley. And we've built a team of really seasoned advisors and also gene editors. With that, I'm happy to take some questions.
Thank you very much. And with this, we are concluding our session. First of all, let me thank to our speakers, because we have seen a wide variety of very interesting ideas, from cancer to ocular diseases to rare diseases. And second, let us also thank, since this was the last session officially, to Leonard Sachs and his associates for organizing this wonderful meeting. I hope you all enjoyed that. And I hope you made many, many contacts, which everybody will profit on. So thank you very much and see you next time.